Good morning, everyone, and welcome to the ELIXIR CZ Republic Friday Coffee, which is the seminar about the tools and databases this infrastructure offered to the users. Molecular biology already touched paleontology or archaeology, and our colleague from the Institute of Molecular Genetics did a great job in collecting the mitochondrial samples from the ancient time. And Edvard Ehler will introduce this database and also the coverage of the genomes they have in their database. So Edvard, floor is yours.
Thank you very much for the introduction. Let me switch the presentation. Today, as it was said in the introduction, I would like to talk about our project that have been already going on for almost 3 years in our Laboratory of Genomics and Bioinformatics at the Institute of Molecular Genetics here in Prague. My name is Edvard Ehler, and on behalf of our whole team, which consists of also Jiří Novotný, Ondřej Moravčík, and Jan Pačes, I would like to talk about this database. Firstly, a short presentation, and then I would take you directly to our web pages, web application, and show you the database. The topic is ancient DNA. When we talk about ancient DNA, the main question maybe would be when the DNA is starting to be really ancient, so we can include it in our research. It is not a set date or set age of the DNA. So in our laboratory, in our database, we have samples, human mitochondrial samples that are the oldest one, about 30,000 years old, so from upper Paleolithic, and the youngest ones are just maybe 150 years old. But generally, the most of the samples that we gather in the database, they are coming from Neolithic times, Bronze Age, and Iron Age. So let's say from the 5th millennium before Christ up until the year 0, just beginning of current era. So this is the main epoch that is targeted now by the human ancient DNA geneticist, but from let's say from animal genetics, we have samples that are much older, even some Neanderthal and early human Homo sapiens samples. They are much older, about 100,000 years. This topic, this research field of ancient DNA is quite broad, I would say, and attracts people of different research specializations. So it is interesting for geneticists, bioinformaticians, of course, population biologists, anthropologists, but also for researchers from historical sciences, archaeologists. Most of our samples, human samples, of course, come from the research and archaeological sites and museums that are curated by archaeologists. The analysis of ancient DNA has two major problems, caveats. The first is degradation. The ancient DNA probably depending on the site where it was deposited and lied there several thousand years. Usually it's very fragmented, and the fragments are experiencing typical damage and patterns of degradation at their ends, different at a 5' end and 3' end. And so this fragmented and degraded DNA, that is what we or people from the ancient DNA wet labs, they have to cope with. The second ancient DNA challenge is contamination, of course, from the soil and from the site. Usually, it's some bacterial contamination. But as I said, most of our samples are coming from the museums and from the some being curated by archaeologists or historians or anthropologists. Some of them are maybe were lying in the museum for the last 80, 100 years. So there were a lot of people doing research on them and touching them and handling them. So modern day contamination is also a problem, which can be sometimes overcome with some specific software tools and protocols, but even a very good sample can be destroyed by, or well, have to be thrown out if the contamination is detected. So let's move to the database itself. I would say the third problem of ancient DNA is the lack of standardization of the data and publication supporting information, and that is the main reason I believe that we have started this database to compile the information about published samples and present it for the research community in some consistent way. For the last 3 years, I've been going through a lot of samples. Currently, we have over 2,300 samples in our database, and most of them had to be prepared for the publication in the database. They had to be prepared by hand. So the hand curation, manually checking the data from different forms of supplementary material published in different journals, and that is probably something that as a I believe most of you are bioinformaticians and maybe structural biologists, geneticists, or molecular biologists. You are probably the databases are already there. So if you are doing a protein research, you just download the proteins in some standard format and just work with it. Nothing like that exists in the ancient DNA field yet. This is something we are trying to do, but the inconsistency of published data is amazing. Well, the sample names doesn't match. The tables are always different. A lot of data and metadata missing. You have to find it somewhere in the text by hand. So that's a I would say that's a big problem and one of the obstacles if you want to get into the field. You have to spend some time, maybe years, just to build up the database of your comparative material or your samples that you want to work on. So, and this is exactly the reason we are working on the database, and we want to upgrade it and continue enhancing and adding new functionalities and new samples. So, sorry. So our goal, offer the data in fair format for all researchers. Nowadays, we are concentrating or providing full mitochondrial genotypes in FASTA format, but even more valuable, I would say, are our metadata. That extra information that we have for every sample in our database. I will show you just in a minute. Another field that we are currently working on is the mitochondrial diseases and disease-causing mutations, their annotation, detection, and we have some tools to help with this. And of course, another new tool, well, not that new, but recently published, is basically a genealogist tool or application of certificate of maternal lineage origin. So let's talk a little bit about our users. The database was started and the first publication and presentation to public was in September 2019. And basically from that time, we have gathered over 20,000 users from all over the world. We have the list of the countries where our users are coming from. So United States, Czech Republic, China, Sweden. It's a very strong labs that are doing research in ancient DNA field, and we are also we have colleagues over there in Uppsala and Stockholm, United Kingdom, Canada, and so on. So as you can see, like every week we have over 100 new and returning users. Clearly on the graph you can see the times where we publish new data, some new functions in the database. So the database updates are very important, and I would say slowly the database is taking off and gathering citations, users, and popularity. So with that being said, what are our plans for the future? Today we have almost 2,500 samples. Maybe it's not that many compared to some genetic or protein database or pathology databases, but the ancient DNA field is quite young. The major articles with using second generation sequencing and large number of samples, they were starting to get published in 2014 or 15, and today I would say there is not more than 5,000, maybe 4,000, 5,000 samples published. So we are gathering them slowly as it needs to be done by hand, and so we have a vast majority of published samples already in our database. The thing is that at least for ancient human DNA or samples, their number is limited, of course. The researchers now are concentrating on Neolithic, Bronze Age, Iron Age, maybe beginning of Middle Ages, but as the number of samples, viable samples, is diminishing, and the new archaeological research is going on, but the samples are scarce. The researchers will start moving into Middle Ages and maybe beginning of modern era samples, and so there is much more samples available in the museum and depositories all around the world. So I'm expecting the number of published samples will rise probably in 5 years horizon, at least one order. So we need to somehow to cope with this and increase the influx of samples into our database. So one of our plans for this is to allow users to enter their own samples in some standardized, foolproof way into our database. That is our plan for future. Uh, as I will show you in a moment, we have several tools, several like sideway applications already running, and we have plans for more. Of course, more users and more Elixir users and cooperation within the Elixir framework. That is our goal for the future years and future grant calls as well. So that is for a short presentation. Uh, I have to acknowledge our colleagues from University Adam Mickiewicz in Poznan. Those are the people that provide quite large part of our samples, and we are cooperating very closely with their wet ancient DNA lab. Anna Juras and Maciej Chilensky. Also, I would like to acknowledge the ancient DNA community and their publication efforts and all the open samples they've published in recent years, and ELIXIR CZ for supporting our database with several internal grants and ongoing support. With that being said, let's move for the database. Okay, so I hope you can see it. So this is the front page. Right away, you can see the statistics of our database: 2,000 samples, 4 continents, 250 cultures. Probably, if you would be a returning user that is already experienced with the database, with just one click here on the front page, you can basically get to the download button, and you can download it all just in 10 seconds maybe and be done with that. If you are first-time user or you're searching for some specific data or specific samples, you will start up the database app itself. Hopefully it will run, and it will start searching. So this is the main application, and you can, as I said, you can download right away, FASTAs, metadata, and so on, or you can search. Let's say you are interested in Neolithic period and want to search for some data from Neolithic. We have over 400 samples from the Neolithic period. You can check their location on this very nice map, and you can see there are several regions, mainly around Europe and Near East, where we got the Neolithic data from. Maybe you would be interested in a little bit refining your search. Let's say you want just Central Europe data, and you are interested in the sequences, so you want data with full mitochondrial DNA. So you refine your search. You can see from Central Europe there are several data. Let's check the Czech Republic. We can switch the map overlay to something more explicit. So we have 3 samples from Kladno area, and we have 2 samples from Prague. Great. Let's have a look on the sample detail page. as you can see, the mapping application is quite nice. You can zoom in almost and see where the sample is coming from. And here on the sample detail page, you can see all the metadata that we are gathering. So identifiers, alternative identifiers, country, region, exact location. So in this sample, it's coming from Kobylisy, Kastírce Street, nice one. Very important part of the metadata, and maybe the most with the most workload together, I would say, is the dating. So the dating is usually in some extra supplementary material, maybe in some completely other article published elsewhere, somewhere differently in different formats. So the dating of the ancient DNA samples is usually done by either C14 dating, as in case of this sample, which is very well dated. You have the calibered data, the uncalibered before present data. You have the laboratory C14 lab code of the sample. That is a very good case, but we have samples that are dated just by archaeological remains or the cultural or artifact that are found around the gravesite. As you can see, we also have the mitochondrial pathologies information for each samples. Well, the graphical presentation is not yet very nice, but all the information is there. Let me show you the sample here. For example, the ones from the Czech Republic. So this is the sample we are looking at, and we can quickly check the pathologies that are or the pathological mutation that are found in this sample. We have several reported mutations detected in this sample. Some of them with conflicting reports. Some of them may be even clinically important. But the status of all those pathologies is reported, which is not the highest status we have. The higher one would be confirmed. So reported status of pathological mutation in our case, in our database, means that it was reported by at least one scientific study. So, and it was not confirmed with another one. So it might be clinically important, but it might not. But we have it here. And talking about mitochondrial pathologies, this week we have just published online our new Metapotto tools, which is quite simple application used for annotate your mitochondrial DNA. So first you have to prepare your mitochondrial sequences in haplograph format, which we have all documents how to do it. Very easy way. You can load an example, and it's a simple tab delimited, basically text table, which you have some sample description, and then you have the mutations that are basically the differences against revised Cambridge reference sequences. And you can search if any of those variants have any pathological phenotype. As we can see from the in sample 1, there were 2 of them found as being potentially pathological, and sample 2. You have another one. You can export them quite easily, export the results, print them, and do whatever you want. So that's our Metapotto tool. And another one I want to show you is this certificate. This is the rationale behind publishing and creating this application is that a lot of traffic and a lot of users we have are coming from the genealogical fields, either genetic genealogy or professional genealogies that are really looking for some specific information. But I suspect that most of the people from this broad genealogical field coming on our side are either amateur researchers or just random people from not from research field, just from any other one that are interested in history, maybe in their family roots. Maybe they have purchased some genealogical surveys from the many companies and services that are viable, and they know their uh, maternal lineage, their mitochondrial haplogroup, and maybe they want to know something more about it. So they we have this certificate application for them. Let's say the name is Mr. Smith. Uh, you know that your haplogroup will be I4A, any other. Let's try this one. And you can basically search your known haplogroup against our whole database, and in this process get this nice certificate that is confirming that we have found, let's say in this case, 9 more people living in different parts of Europe, living in different periods. Some of them are coming from Czech Republic, Neolithic, Bronze Age, Copper Age. Some of them from Germany, Lithuania, Great Britain, Poland, Russia. So you can basically get some idea about where your maternal lineage is coming from, where did the people from the same lineage, where did they live, maybe where did they come from, and basically what is their distribution. I4A is an interesting haplogroup that is quite low frequency in Europe, around 3-4%, but it's located usually in like the most common people and ethnic groups or cultural groups carrying this mitochondrial haplogroup nowadays are the people from this region of Slovakia, Eastern Poland, Belarus, and Western Ukraine, called Lemki people, or Lemkos in Czech, Lemkové. So this is the most frequent group of today's Europe that carries I4A, around 11 or 12% of Lemkis carrying I4A. So this is the certificate, and let's close this page and have a quick look at the documentation. So all the metadata, all the gathering, reconstruction of DNA sequencing, our sources, reference articles, and so on. All is quite well covered. I have to say we have the brief description of the bioinformatic pipeline for the mitochondrial sequences reconstruction. We have description of the tool, of the certificate, and we have let me show this because this is a very nice page and functionality. We have all the scientific articles that we gather data from one page, and which can be used quite easily. For example, you would be interested in Damgard 2018 article, which published over 130 new samples from Eurasian steppes. So you can very quickly filter these data and have a look at that. And you can see the steppe people from one article published and displayed one map, and you can just download or select the ones that you want and quickly download them and work with them and do whatever you want. So, with that being said, I would like to end my presentation and introduction of our database, and I hope you will be maybe interested in our database and might use it or recommend it to use in the future. And again, I would like to thank to ELIXIR for supporting us, and I'm ready for questions.
Thank you, Ed. We have a few questions here, so I try to make it very short and also expect brief answers. So who are the users of the database, and are there publication or study based on the data you provided?
Yes. Yes, there are several publications. With some of them we have here two of our articles, but the main source article is our NAR database issue article. This is the source article, and so far it has, I would say, 7-8 citations from different groups all over the world, slowly gathering more.
Okay, thank you. Another question is, which persistent identifier scheme do you use?
I'm sorry, I didn't understand which.
Persistent identifier scheme do you use? Is a question from Robert Pergel. Probably Robert will explain what he meant.
Okay.
Yes. Hi, Edvard. So you mentioned that you are trying to be fair. So the question, the first thing to be fair is to have a persistent identifier scheme, and this is quite tricky. So I'm interested, which one do you use, and what's your experience? But there's no time for that. So just which one do you use?
I'm not sure now. I don't think we use one, but yeah, we use the well, of course, unique identifiers for each sample, but I'm not sure we are using this frameworks of persistent identifiers.
So Robert also suggests that this would be a great to cooperate with you to verify all the data. So probably it's a discussion between Robert and you can actually go ahead in that verification of the data. So another question here is, what is a typical sequencing coverage of the ancient DNA samples?
Well, the coverage, I have to say, if it's 3, is good. You can filter the database also for coverage. It is not uncommon for the ancient DNA samples to be coverage under 1, I have to say, but the usual coverage of the good samples is between, let's say, 2 and 5. But we have the tool here. You can choose your coverage. Let's say samples that have coverage at least 1. You can submit and see. how many. But the higher coverages are rather rare. So between 2 and 5, I would say.
Okay, so last question is quite long. It's from Martin Mokresh. I tried to read it slowly, so you didn't even mention how you process the data. The aDNA isolated supposedly contains mostly gDNA contamination. So how do you get around genes encoded also in gDNA? Do you emphasize in the database and website genes and mutation which could be caused by mismapped reads?
Okay, so we have the sequences from different sources. The ones that we reconstruct, we have some contamination basically tools, or we are checking using the map damage. So we are checking that the fragments we are using to reconstruct the mitochondrial DNA are really ancient DNA. Usually we also use the Schmutze algorithm to check for the modern day contamination. But part of the data, if the researchers will enter their mitochondrial DNA into GeneBank, we will just download those sequences from GeneBank, and we don't check for any additional contamination or DNA damage or DNA quality.
Okay, so time is running. Thank you, Edda, once more, and thank you also to all of you for listening. And see you next week on Friday. And it will be about the galaxy and usage of the galaxy in the molecular biology. So have a good weekend and see you next Friday. Bye.
Thank you very much. Bye-bye.